This is Ananya Pandey, and today I am going to teach about the cell, structure and its functions. Now we know that the things around us are either living or non-living. We also know that the living beings carry out certain functions to survive. Now to carry out those functions we need different set of organs, and these organs are made up of different kind of cells, the assembling of different kind of cells. The cells may be compared to the bricks. Just like bricks are essential to make a building, similarly the cells are essential and assembled to make the body of every organism. So we can call the cell as the fundamental unit of life. It is a self-replicating structure. Now it was Robert Hooke in 1665 who observed the slices of cork under a simple magnifying device. This is a simple magnifying device that he used to observe the slices of cork. This is cork. Cork is a part of the bark of a tree. Okay. So he observed this under the microscope. This is the hexagonal shaped thing that he observed, and he named it as cell. Now what he observed was that there was no movement in these cells. So he declared that these cells are dead cells. Later, Anton von Leeuwenhoek, he observed cells under another compound microscope with higher magnification, and here the cells exhibited some form of movement in them. There was mobility, and as a result Leeuwenhoek, he concluded that these microscopic entities were alive, and he named these entities as animalcules. So he observed that the cells are living, and he used the compound microscope. They were development in the microscope also, and when they were development in the microscope we could observe the clear picture of how the cell looks like and what are the different properties of the cell. Later in 1883, Robert Brown, he was a Scottish botanist, he provided the very first insights into the cell structure, and he was also able to describe the nucleus, the structure of nucleus present in the cells of orchids. So he gave a detailed picture of the cell structure, and he also mentioned about the structure of nucleus which he observed in the cells of orchids. Now next we have to talk about the cell theory, which was formulated by M.J. Sheldon and Theodore Schwann in 1838. What they maintained was the first, all organisms are composed of cells. Number one. Number two, that cell is a structural and functional unit of life. And number three, that cells arise from pre-existing cells. This third point was mentioned by Rudolf Karl Virchow. Okay, the first 2 points were mentioned collectively by M.G. Sheldon and Theodore Schwann, but to add another point and very important point to this cell theory, that cells arise from pre-existing cells, was done by Rudolf Karl Virchow. Now to give a brief picture about the cell theory timeline, here is the clear picture that you can see. We have already discussed about 1665 Robert Hooke, who he termed the word cell for describing biological organism, because he said that they resembled monk's cell. Then in 1674, Anton von Leeuwenhoek, he is also known as the father of microbiology, and he was the first person to ever see microorganism. I have already mentioned that he named them as animalcules, and he has contributed towards the establishment of microbiology as well. In 1827, Robert Brown, he is known for discovering the cell nucleus, and he gave one of the earliest detailed description of the cell nucleus, and where he observed it in the cells of orchids. Okay, and 1839, Jacob Sheldon and Theodore Schwann, they propounded the cell theory, and they were both like co-founders of the cell theory, and the three principles of cell theory that we have already discussed were that all organisms are composed of cell. Cell is the structural and functional unit of life, and cells arise from pre-existing cells. Then 1855, Rudolf Karl Virchow, he gave the third principle, that is the cells, they arise from the pre-existing cells. The existing cells divide to form new cells. And finally in 1855, Louis Pasteur, He made significant discoveries in chemistry, including treatments and research of many diseases. So, this is a brief picture of the cell theory timeline. But there is one exception to the cell theory, that is virus. Viruses are always a conflicting topic. Viruses are considered alive by some because they enter the cell and they become alive, but they're not made up of cells. Viruses have many features of life, but according to the cell theory, they are not alive. Okay, so this is a brief timeline, or you can say the history of cell theory, how different botanists, microbiologists have given their views on cell. Now, how can you define a cell? A cell may be defined as a unit of protoplasm bound by a plasma or cell membrane and possessing a nucleus. Now, what is a protoplasm? Protoplasm is the life-giving substance and includes the cytoplasm and the nucleus. So, protoplasm is what? Cytoplasm and nucleus. And a cell may be defined as a unit of protoplasm. Okay. Now, different substances that make a cell collectively called protoplasm, and it consists of water. Majority of water is present in the cell, that is 70 to 80%. Then there is carbohydrates, lipids, proteins, electrolyte, sodium. Electrolyte contains sodium, potassium, magnesium, calcium, phosphate, chloride, and bicarbonate. So this is what protoplasm consists of. Now, according to the number of cells present, the organisms are divided as unicellular organism and multicellular organisms. The unicellular organisms, as you can understand from the name itself, uni means one. So, organisms made up of only one or only a single cell. So, example can be amoeba and paramecium. Now, second is multicellular organisms, or multi means many or more than one. So, organisms made up of more than one cell. Example, human, cow, and rose, etc. Now, according to the shape of cells also, we have classified different kinds of cells. So, you can see here, these cells are rod shaped, which are called as bacillus. This is spherical in shape, and this is called coccus. And this is spiral in shape, which is called spirillus. You can see this is in comma shape, which is called vibrio. Now, in humans also, we have different shape of cells. You can see here RBC, which is round and biconcave. This is round and this is biconcave. The nerve cells, which is the branched and really long cells. You can see how many branches are there here, and this is pretty much long. So this is nerve cell. You can see the white blood cell, which is in shape of amoeba. So it is called amoeboid. Here it is columnar epithelial cell, which is long and narrow. Okay. Now here is guard cells, which you can see in stomatal pose. Okay, this is kidney shaped. So these are the different shape of cells that are present in different organisms. Okay. Now there are two types of cells majorly. One is prokaryotic cell, another is eukaryotic cells. Now what is pro and what is eu? Firstly, we have to understand this. Pro means which is early, which is primitive, and eu means true, and karyon means nucleus. So with the name itself, you can understand the next point, which says that in prokaryotic cell, the nucleus is not well defined and it is known as nucleoid. And in eukaryotic cell, the nucleus is well defined and it is surrounded by a nuclear membrane. In prokaryotic cell, there is a single and circular chromosome, and in eukaryotic cell, it possesses more than one and linear chromosome. Prokaryotic cell, here the membrane-bound organelles are absent. Basically, there are no membrane around the organelles and there is no well-defined nucleus. And here in eukaryotic cell, membrane-bound cell organelles are present, is well-defined. The example of prokaryotic cell can be a bacterial cell and eukaryotic cell can be human cell. So in the next slide, you can get a clear picture of how eukaryotic cell and prokaryotic cell looks like. So here you can see in eukaryotic cell there is a nucleolus and it is membrane enclosed. And in here prokaryote you can see a nucleoid. There is no well-defined nucleus and there is no nuclear membrane. There is no cell membrane. Okay, so this is what eukaryotic and prokaryotic cell looks like. Now next we have to discuss about the cell structure. The cell structure comprises individual components with specific functions essential to carry out life processes. Now, the different components that include in a cell are cell wall, cell membrane, cytoplasm, nucleus, and cell organelles. Okay, we'll discuss about this in detail. The subcellular structure that we have already discussed is cell wall, cell membrane, cytoplasm and its organelles, and nucleus. We'll discuss it one by one. The first and foremost is the cell wall. Now, cell wall is only and only present in the plant cell structure. It is made up of cellulose, hemicellulose, and pectin. Now, cell wall is present exclusively in plants, and I've already mentioned it protects the plasma membrane and other cellular components. Also, it is the outermost layer of plant cells. It is a rigid and stiff structure surrounding the cell membrane. It provides shape and support to the cell and protects them from mechanical shocks and injuries. Now, the second we have to discuss about cell membrane. It is a thin, pliable, elastic outermost structure which envelops the cell. It protects the cell, it is present in both plant and animal cell. It protects the cell from external environment. Also, it is present in all, like plant and animal cells, both of the cells has cell membrane. It controls the movement of substance in and out of the cell. It has pores in the membrane which, controls the movement of substance in and out of the cells. The next we have to discuss about cytoplasm. Cytoplasm is present inside the cell membrane. It is a thick, clear, jelly-like substance, and most of the chemical reactions within the cell take place in the cytoplasm. It is a fluid where all the chemical reactions happen. It is bounded by a plasma membrane, and all the cell organelles like endoplasmic reticulum, white vacuoles, and all of the cell organelles are suspended in the cytoplasm. Okay. Now, cytosol. The cytosol is the aqueous part of the cytoplasm. It is outside all the organelles and also contains distinctive proteins. It is 70% of the total cell volume is cytosol. Okay, it has many chemical elements in the form of atoms and ions and molecules. Okay, the next is nucleus. The nucleus contains the hereditary material of the cell, the DNA. It sends signals to the cells to grow, mature, divide, and die. Now, nucleus is surrounded by a nuclear envelope that separates the DNA from the rest of the cell. Now, the nucleus protects the DNA and is an integral component of a plant cell structure. Okay. Now, coming to the cell organelles that I've already mentioned, the different cell organelles that are present in the cytoplasm are mitochondria, endoplasmic reticulum, lysosome, Golgi apparatus, chromosome, ribosome, and vacuole. Now, each organelle is bounded by a lipid membrane. Okay, so every organelle is bounded by a lipid membrane, and every cell organelle has specific function assigned to them. First, we will discuss about the mitochondria. So, the mitochondria were first observed by Kolliker in 1850 as granular structures in the striated muscles. Okay, the mitochondria are called a powerhouse of the cell. The structure, the length and diameter you can see here. It is filamentous or globular in shape. As you can see in this picture, it is filamentous and globular in shape. It has an inner membrane and outer membrane. This is called a cristae, and it has a fluid filled in it. This sac are called the cristae, and it has fluid in it which is called matrix. And this is a filamentous or globular in shape. Now, the functions of mitochondria, as you can see here, is that this is the power generating units of the cell. It is important to maintain proper concentration of calcium ions within the various compartments of the cell. And here the energy transduction is through respiration. The role of mitochondria is most important in respiration, and the energy transduction happens here through respiration, and this is responsible for thermogenesis. Now, the next is endoplasmic reticulum. So, endoplasmic reticulum, you can see this is tubular and kind of vesicular structure that you can see here, and it is suspended in the cytoplasm. Okay, so there is a network also. There is a network of tubular and flat vesicular structure in the cytoplasm. The extensive network hota hai. Then this is called cisternae. As you can see here, this is tubular and vesicular structure which are called cisternae. The spaces filled inside the tubule is filled with endoplasmic matrix. These spaces is filled with endoplasmic matrix. Then there are two types of endoplasmic reticulum. The one is smooth endoplasmic reticulum and another is rough endoplasmic reticulum. Now, smooth endoplasmic reticulum, here it is ribosomes absent, and in rough the ribosomes are present. Now, smooth site of synthesis of lipid and steroid hormones. So, lipid and steroid hormones are synthesized here, and the rough endoplasmic reticulum contains the site of proteins, and here it is protein is synthesized, processed and packaged. Okay. Now, the smooth endoplasmic reticulum is mainly present in lipid forming cells like adipocytes. These are lipid forming cells. Interterrestrial cells of testis, glycogen storing cells of liver, adrenal cortex cell, muscle cell, leukocytes. And rough is mainly present in protein forming cells such as pancreatic echinal cells, goblet cell, antibody producing plasma cells, Nisc granules of nerve cells, etc. So these are the two types of endoplasmic reticulum. The function, what are the functions of endoplasmic reticulum? Yeah, here it is that it helps in synthesis of proteins. It also segregates proteins. Here also the fatty acids are unsaturated. It helps in muscle contraction. Endoplasmic reticulum is commonly known as sacroplasmic reticulum in muscle fibers because it helps in muscle contraction. And mainly their function is synthesis of protein, and they segregate protein also. Now the next comes the Golgi bodies. Golgi bodies is a collection of membrane enclosed sacs composed of 4 more stacked layers of thin flat enclosed vessels lying near the side of nucleus. So it is also membrane enclosed sacs which are composed of 4 or more stacked layers of thin flat enclosed vessels. So it has multiple discrete compartments. It's layered very much. Stacked layers होती हैं. Thin flat enclosed vessels, and consists of 4 functionally distinct regions. The one is the cis-Golgi network, second is the Golgi stack which is divided into the medial and trans subcompartments, and the third is the trans-Golgi network. Now this is the Golgi apparatus. You can see this is cisternae. The folded structures basically are called as cisternae. And there is lumen, the sac that contains it is called lumen. Now this is the rounded structure are called secretory vesicle which are at the lower end, and the upper end it is incoming transport vesicle. The upper end is called the cis phase and the lower end is called the trans phase. You can see here is the 2D structure and the 3D structure. You can get a clear picture of how Golgi apparatus or Golgi bodies looks like. Okay, so next we have to see the functions of the Golgi apparatus. So Golgi apparatus helps in the wrapping and packaging department of the cell. It is basically the wrapping and packaging department of the cell. It produces secretion granules, that is membrane enclosed complexes which store hormones and enzymes in the protein secreting cells. It packages proteins. Now, site of formation of lysosome. It is the site of formation of lysosome. Lysosomes are large irregular structure. We will study about it also in the next slides. It is a large irregular structure surrounded by membrane which are present in the cytoplasm. It adds certain carbohydrates to form glycoproteins which play an important role in the association of the cells to form tissues. Now, we mentioned about lysosome. So here it is the lysosome. The lysosome are the irregular structure which is surrounded by a unit membrane. This is the unit membrane. This is the lipid layer. And this is hydrolytic enzyme mixture. It is more acidic than the rest of the cytoplasm. External bacteria as well as worn out cell components are digested in them. They are so acidic that the worn out cell components are digested in them. They are also known as the suicidal bags of the cell. Now why they are known as the suicidal bags of the cell, we will get to know in the next slide. Now the function of lysosome, it acts as a form of digestive system of the cell, because enzymes present in it can digest essentially all macromolecules. It engulfs the worn out components of the cells. It engulfs exogenous substances like bacteria and degrade them. Now when a cell dies, the lysosomal enzymes cause autolysis of the remnant. The remnant remains in the cell, in the cytoplasm. The lysosome digests all of it. So that is why lysosomes are referred to as the suicidal bags. Now the next we have chromosome, ribosomes and vacuoles. Chromosomes play a crucial role in determining the sex of an individual. Now each human cells contain 23 pairs of chromosomes. Now ribosome, these are the protein synthesizers of the cell. Here the protein are synthesized. Next is vacuoles. The vacuoles are basically the storehouse. Why this is a storehouse? Because it stores food, water and other waste material in the cell. Now next we have to understand the difference between a plant cell and animal cell. So here you can see that plant cell, there is a cellulose wall that is present external to the cell membrane. We have already discussed when we have talked about the cell wall, and it is only present in the plant cell. While in animal cell there is no cell wall, outermost structure is cell membrane or plasma membrane. In plant cells the vacuoles are usually large, very large, and in animal cell it is generally absent, and if present that is usually small. Okay, plant cell has plastids present and animal cell plastids are absent. In plant cells the Golgi body are present in the form of units known as dictyosomes, whereas in animal cell the Golgi body is well developed, having two cisternae. The cisternae is the folded structure that we have discussed while we were discussing the Golgi body. Where the plant cell, here the centriole is absent, whereas the animal cell it is present. So here is the picture that you can see of the plant cell and animal cell. That is all about the cell, the structure and its functions. Hope you've liked the lesson that I have taught. Thank you so much.